So yes, now we have the concept of cloning vectors. Plasmids and bacteriophages have the ability to replicate within the bacterial cells independent of the chromosomal DNA. So a plasmid can replicate without the control of the chromosomal DNA. The question is, what should be a good vector? एक plasmid और एक vector अगर यूज कर रहे हैं, what are the characteristics of the vectors that are important for DNA recombinant technology? सबसे पहले features that are required to facilitate cloning in a vector, origin of replication or ora. तो सर ये है बैक्टीरिया and ये है बैक्टीरिया. Within the bacteria we have its own DNA. This is the circular bacterial DNA which is attached to the cell membrane. It is attached to cell membrane, and this is your freely circulating DNA called as a plasmid. So sir, ये plasmid, when will it be accepted by the bacteria and when will it replicate? बैक्टीरिया के अंदर एक सीक्वेंस होता है which is called as the ora, which is called as origin of replication. And origin of replication को बहुत सारे enzymes recognize करते हैं, and these enzymes recognize the ori, and then using the ori the bacterial DNA undergoes replication. The same sequence or ori should be also present in the plasmid. Plasmid needs to have same sequence of ATGC or nucleotide sequence, जो same nucleotide sequence which is origin of bacterial replication, same sequence has to be there called as ori even in case of plasmid. Then the enzymes will recognize not only ori of the bacterial DNA and make it replicate, enzyme will recognize ori of the plasmid, and the plasmid will also replicate. And the ori is such, it will support a high copy number. So enzymes recognize ori of the plasmid and make multiple plasmids. तो बहुत सारे plasmids बनाएगा, which means it has got a high copy number. So ori has to be matching, and ori should support a high copy number of the plasmid. And along with the ori matching, the cloning sites, the vector needs to have, or plasmid needs to have very few, preferably single recognition sites for commonly used restriction enzyme. एक restriction site होना चाहिए, तो समझो PBr322. PBr322 is an artificial plasmid which is the most commonly used plasmid for DNA recombinant technology. It has got ori, which is the origin of replication, and it has got in its plasmid lots of recognition sites. So here we have EcoR1 का recognition site. If you remember, what is the EcoR1 recognition site? GAA-TTC, and यहाँ पर भी है GAA-TTC. And similarly, EcoR1 का recognition site ये है. Similarly, for Hind3 there is a recognition site, for BAM-H1 there is a recognition site, for SAL1 there is a recognition site, for PBU2 there is a recognition site, for PSC1 there is a recognition site, for PBU1 there is a recognition site. तो बहुत सारे restriction sites है for different enzymes, but mind you there is a single recognition site. Say for EcoR1 you have one GAA-TTC which will be cut. If the same sequence was present here also, and the same sequence was present here also, then EcoR1 जब आप डालते थे PBr322 पे, it would cut here, it would cut here, it would cut here, and the entire plasmid will become multiple fragments which will complicate the process. तो सर आपके पास रिकग्निशन साइट होना चाहिए, पर आपके पास मल्टीपल रिकग्निशन साइट नहीं होने चाहिए, otherwise the restriction enzyme will cut it at multiple sites and the plasmid will become fragments. So you should have few, preferably single recognition site for the commonly used restriction endonuclease enzyme. And important, there should be selectable markers. Selectable markers is addition to the ori, selectable markers होने चाहिए which identify and eliminate non-transformers and selectively permit the growth of transformers. तो एक marker gene होना चाहिए which will help you in identifying whether the plasmid has been accepted and is forming multiple copies or the plasmid has not been accepted. So selectable markers, what are these? What do you mean by these, sir? Sir, normally genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin are selectable markers for E. coli. So antibiotic resistance genes are the selectable markers for E. coli. For example, समझो आपके पास है PBR 322, तो ये है PBR 322, उसके अंदर you have one gene which is called as ampicillin resistance gene, another gene which is called as tetracycline resistance gene, and ampicillin resistance gene and tetracycline resistance gene. So sir, समझो this is PBR 322 and it has got ampicillin resistance gene and it has got tetracycline resistance gene. Now these two genes will undergo transcription translation to form enzymes which will make the plasmid become resistant to antibiotic ampicillin and tetracycline. Suppose you have only E. coli, and only E. coli without plasmid, तो उसमें plasmid नहीं है, you put over it ampicillin, the bacteria will die. You put over it tetracycline antibiotic, the bacteria will die. But समझो you put in the bacteria PBR 322, it has got ampicillin resistance gene and tetracycline resistance gene. And now suppose the ori of the bacterial DNA and the ori of the plasmid are matching. If the two oris are matching, then enzymes will help in recognizing the ori, and not only the bacterial DNA will replicate, even PBR 322, the plasmid will replicate, and the plasmid will form multiple copies of PBR 322, and they will have ampicillin resistance gene, tetracycline resistance gene, ampicillin resistance gene, tetracycline resistance gene. And now suppose you put over it ampicillin or tetracycline, then in the presence of ampicillin resistance gene, the bacteria will survive. And you put tetracycline, then because of tetracycline resistance gene, the bacteria will survive. This means successful transformation has taken place. इसे क्या कहते हैं? Successful transformation. In other words, the plasmid has been accepted by the bacteria, and along with the bacteria, the plasmid is also undergoing replication, and a successful transformation has taken place, which means the bacteria has undergone transformation. Now, you can do transformation, and now you can use that plasmid for DNA recombinant technology. तो इसे कैसे आप मार्कर की मदद से मेक आउट करेंगे? Suppose you have this PBR 322. Bacteria has accepted ori of the bacteria and ori of the PBR 322 are matching. Means along with bacterial replication, the plasmid is also multiplying, and ampicillin resistance gene provides resistance to ampicillin. Tetracycline resistance gene provides resistance to tetracycline. Means the bacteria has accepted the plasmid. Transformation has successfully taken place. Now we use an enzyme called BAF-H1, and BAF-H1 say we make a cut. Where is the restriction site for BAF-H1? The BAF-H1 restriction site is inside the tetracycline resistance gene. So we use BAF-H1, make a cut in the tetracycline resistance gene, and now we put our gene of interest over here. Our gene of interest, say gene for insulin, हमने यहाँ डाल दिया. Now that we have put in the gene for insulin, and this is a chimeric DNA or a recombinant DNA, and this chimeric DNA or recombinant DNA is now put back into the bacteria. So ये है bacterial DNA, and this is plasmid PBR 322. This is ampicillin resistance gene. This was tetracycline resistance gene, but in the tetracycline resistance gene, we have put the gene for insulin. So this is gene for insulin, means the the plasmid has undergone recombination. And now, what do? You put over it ampicillin, then ampicillin resistance gene will make the bacteria resistant to ampicillin. But you put over it tetracycline, then what will happen to the plasmid? Well, tetracycline resistance gene, we have caused its inactivation. We have caused its inactivation by putting in the gene for insulin, and because we have caused inactivation by putting in gene for insulin, excuse me, all of you.